everyone, and welcome to this explainer. Today, we are diving headfirst into the absolutely fascinating world of chiral chromatography. We're going to figure out exactly how scientists pull off what seems impossible, separating microscopic chemical twins. It's this brilliant mashup of chemistry, physics, and some seriously incredible molecular engineering. So right off the bat, here's the core puzzle we're tackling today. How in the world do you tell apart identical-looking molecular twins? Think about a pair of gloves, right? They look basically identical, they're made of the exact same leather or cotton, and they weigh the exact same amount. But try putting your left hand into the right glove, and you instantly know they are fundamentally different. Standard chemical separation methods, they just see two identical gloves. They can't tell them apart because the physical properties are exactly the same. So how do we separate them? Well, to make sense of all this, here's our roadmap. We'll quickly cover 1. The molecular twin problem. 2. Principles of chiral chromatography. 3. Measuring our optical purity. 4. Early solutions, Perkel columns. 5. The cyclodextrin revolution. And finally, 6. Reversible complexes in action. Okay, let's jump right into section 1, the molecular twin problem. When we're talking about these twins, the actual scientific term is enantiomers. These are pure compounds that contain a center of asymmetry, which basically means they exist in two distinct forms, which we usually call the R and S enantiomers. They are perfect chemical mirror images of one another. Now, in a lab beaker, they look and act practically identical, but separating them is absolutely crucial. Why? Because when these R and S forms get introduced into highly asymmetrical environments, like, say, the human body, they can behave completely differently. Moving right along to section 2, principles of chiral chromatography. So how do we actually pull off this separation? The secret weapon here is something called a chiral stationary phase. Remember that glove analogy? If standard chromatography is like trying to sort hands just by measuring how much they weigh, chiral chromatography works by actually providing a molecular glove. If the stationary phase inside our chromatography column is entirely made up of, let's say, right-handed gloves, then only the right-handed molecular twins are going to fit perfectly. They get held back. Meanwhile, the left-handed twins, they just slip right on through. It's this highly selective interaction that allows us to finally separate them and observe two distinct peaks. And when we run this process, the results are really beautiful. Instead of getting one massive, confusing blob of an elution peak where both twins rush out at the exact same time, our stationary phase slows one twin down more than the other. What we get is a perfectly clear chromatogram displaying two distinct peaks, one representing our R enantiomer and the other representing our S enantiomer. Which brings us to section 3, measuring our optical purity. Okay, so once we've successfully separated those peaks, we need a reliable way to mathematically prove it. We measure what's called optical purity, which is defined in terms of enantiomeric excess, or EE percentage. The math here is surprisingly straightforward. We basically just look at AR and AS, which represent the total area under each of those two peaks we just talked about. Since the area of each peak is directly proportional to how much of each form is actually in there, doing a quick calculation with those areas tells us exactly how pure our separated sample really is. Next up is section 4, early solutions, specifically Perkel columns. So how did chemists first try to build these molecular gloves? The early pioneers came up with what we call Perkel columns. They took a solid silica support base, added some bonded aminopropyl groups, and used that to anchor a specific derivative of D-phenylglycine. And honestly, it was a totally brilliant proof of concept. But it had some pretty major flaws. For one, these phases were relatively unstable over time. On top of that, their selectivity coefficient was painfully close to 1. In plain English, that means the physical separation between the two twins was barely noticeable. It worked, but the scientific community desperately needed something better. That takes us to section 5, the cyclodextrin revolution. When we compare these two eras of technology, it's a literal night and day difference, a total revolution. While the old Perkel columns really struggled with that instability and low selectivity, the introduction of cyclodextrins changed the game overnight. Cyclodextrins are optically active oligosaccharides, basically complex sugars. And instead of those fragile setups from the past, these bad boys are securely bonded to the silica gel using a sturdy little hydrocarbon chain linker. The result? A highly stable, incredibly selective stationary phase. Let's actually zoom in and build this modern marvel piece by piece. You start with your foundation, which is a silica gel bead. Attached to that is your small hydrocarbon chain linker arm. But the cyclodextrin molecule itself is the real star of the show. The outside of it is hydrophilic, meaning it loves water. But the inside, it forms a hydrophobic, water-repelling internal cavity. And here's the wildest part. That internal cavity is exactly zero point 0.8 nanometers wide and 0.8 nanometers high. We are literally talking about engineering a sub-nanometer hydrophobic bucket to catch very specific molecules. That's just wild. Finally, we've made it to Section 6, Reversible Complexes in Action. So let's see how this microscopic zero point 0.8 nanometer bucket actually separates a racemic mixture. As a bunch of compounds, including our identical molecular twins, flow past, they approach the cyclodextrin. Because of the super specific shape of that tiny cavity, the molecules experience selective inclusion. One twin is going to fit perfectly into that hydrophobic pocket, while its mirror image twin is just a bit too awkward to fit in the exact same way. This interaction forms diastereoisomers at the surface, creating reversible complexes. Basically, one twin gets temporarily caught in the bucket, while the other just keeps flowing right on by. And just like that, our completely identical twins are separated into those distinct peaks. Now, you might be asking yourself, so what? Why go to all this trouble? Well, what you absolutely need to take away from this is that separating RNS enantiomers is not just some neat party trick for chemists. It literally saves lives. Take the separation of a racemic mixture of an amino acid, like DL-tryptophan. Because our own human bodies are inherently chiral, meaning we're full of our own asymmetrical locks and keys. One molecular twin might be a life-saving medicine, while its exact mirror image could be totally useless or even highly toxic. Mastering tools like cyclodextrin phases guarantees that we are isolating and delivering only the beneficial version of a molecule. It's honestly a staggering testament to human ingenuity. The fact that we can reliably manipulate chemistry at a scale of 0.8 nanometers to create safer medicines is just incredible. And if a microscopic sieve this tiny and precise can separate exact chemical twins, it really makes you wonder, what other microscopic sieves and structural marvels are just waiting out there to be discovered? It's a fascinating thought to leave you with today. I really hope this explainer sparked your curiosity and helped to demystify the amazing world of chiral chromatography. Keep exploring, keep asking questions, and we'll catch you next time.